Hey guys, let's go through the steps of a skeletal muscle contraction. We're going to start by drawing the end of a neuron called the axon terminal. It looks like this, and it has a couple pores where the neurotransmitter is released. Remember, a neurotransmitter is a chemical made by a neuron, a nerve cell. That's it. And this, of course, would be the axon terminal of a neuron. So a neuron, and this would be the axon terminal. And then between the neuron and the muscle cell is a space called the synapse. It's a very important space. And so we need to make sure to include that. The synapse is the space. And then below that, we're going to draw a skeletal muscle cell. And we're just going to draw it something like this, a little simplified. But this is a skeletal muscle. So remember, this is going to be attached to a bone. It's a little bit different in a cardiac cell and a smooth muscle cell. So in the neuron, there is it's making a neurotransmitter. In this case, it's making acetylcholine. Acetylcholine is going to be the main neurotransmitter in a skeletal muscle contraction. Acetylcholine is made up of two parts. It's made up of an acetyl unit, which comes from the mitochondria, and a choline, which comes from your diet. It's found very high in eggs, things like that. So things that are going to be really helpful for the functioning of your muscle system and your nervous system. Eggs is a wonderful thing to eat. It's also going to be found in some other foods. But your acetyl, that's going to come from the mitochondria. Mitochondria uses it for things like the Krebs cycle, also donates it to make acetylcholine, and choline comes from your diet. So we'll say from the diet, right? And it is put together by an enzyme called CHAT. That's an acronym. It's a really long name. You just need to know the acronym. So we're going to say it looks something like this, and we'll say the acetyl looks like a circle and the choline looks like a triangle. And this CHAT glues them together. So it makes a bond between the acetyl and the choline, and it makes acetylcholine. So here we have our acetylcholine, which we're going to shorten to capital A, capital C, lowercase h for acetylcholine. It's just a shortening way. So in the neuron, we have our CHAT enzyme making acetylcholine. And the acetylcholine is getting put in a vesicle. The cell likes to put the neurotransmitters in vesicles, and they're going to sit there until it gets a signal to be released out of the cell. And so that's what we're going to do. We're going to send a signal from the brain to the neuron to tell the muscle cell to contract. And if you've ever heard that a nerve cell sends an electrical signal, the electrical signal is sodium ions. Sodium ions have a positive charge, so it's going to carry a charge along it. So we have this wave of sodium ions that's coming all the way down the neuron. It's traveled all the way down the axon, all the way to the axon terminal here. And so we have sodium ions that are traveling down, and the cell at rest is pretty negative. So the cell at rest is going to be like a negative 65 millivolts, right? And so when the sodium ions come in, it gets more and more and more positive until it gets to positive 40. And at positive 40, that's a very important number to know because we're going to see that same number when we get into the nervous system. We're also going to see the same number in the muscle cell. At positive 40, it opens a voltage-gated channel. What that means is the channel opens at a certain voltage, in this case, 40 millivolts. And the channel that opens is our calcium channel. So the calcium channel over here stays closed until until it reaches the charge. The cell starts at rest. All the calcium is outside the cell. And it doesn't want to be there because there's a bunch of calcium there. It doesn't want to hang out with other calcium. So it very much wants to go inside the cell, but it can't because the gate is closed. So as soon as this cell reaches positive 40 millivolts, that gate opens, This calcium is going to rush into our axon terminal here. So all of our calcium comes in. And the thing about calcium is that calcium in the body moves things. In the neuron, it's going to move the neurotransmitter. In a muscle cell, it's going to move our muscle filaments, and we'll get to that in a second. So the calcium comes and it binds to this vesicle containing the neurotransmitter acetylcholine, and calcium makes things move. So it makes the vesicle move to the cell membrane of the neuron. So it's going to move here, and it's going to fuse with the cell membrane. But as you know, things cannot cross the cell membrane very easily. The cell doesn't want things coming in and going whenever it wants. It tightly controls what enters and leaves the cell. And so to help things leave the cell, we have the help of proteins. In this case, the protein that helps it leave the cell is called SNARE, also an acronym, all caps. And so SNARE looks like this. I say it looks like the nose hairs in the nose. And this is SNARE. SNARE is also the pharmaceutical target for Botox. Botox cuts SNARE, and that's why people are not able to contract the muscles of their face, because their SNARE was broken. So the neurotransmitters never released to tell the muscle cells to contract. Kind of interesting connection there. So the SNARE helps the acetylcholine move across the cell membrane and leave the cell. That's called exocytosis. Okay, so our acetylcholine has now left the neuron and it's out in the synapse. And this neuron is not just releasing one acetylcholine, it's releasing millions of them. We're simplifying it, right? That way that we can kind of see it all on one diagram. So on the surface of the skeletal muscle cell, there's receptors. These are acetylcholine receptors, and I'm going to draw them like this. This is not exactly what they look like, obviously, but we're going to draw them like this along the skeletal muscle cell. The acetylcholine is going to diffuse across the synapse. In reality, it's a very, very, very tiny synapse. We're drawing it very big, right? But it goes and it's going to attach to the acetylcholine receptor and activate it. This receptor is a ligand-gated channel. So what that means is when a ligand, in this case acetylcholine, binds to it, it opens a channel for an ion. And that ion is the same one we saw in the neuron. So you're going to see the same things repeated, because when the body finds something that works, it copies it and repeats it because it works. So the acetylcholine is going to bind to the receptor and it's going to activate it. We'll talk about that. We'll talk about that in a second. First, I want to take a quick side note and explain the enzyme acetylcholinesterase. Acetylcholinesterase is a long name, but all they did was took the word acetyl and added esterase. So it's an enzyme that breaks down acetylcholine, right? Don't let it confuse you. Acetylcholinesterase is all in the synapse, and it goes through and it breaks the bonds of acetylcholine. Why would you want to break something that you just made? Well, every time acetylcholine touches a muscle cell, it contracts. So you don't want it to stay in the synapse and keep touching the muscle cell, or the muscle cell would keep contracting, which is spasms or tremors. Not good. So as soon as the acetylcholine signals the muscle cell, we want to get rid of it. So that's acetylcholinesterase. I like to draw it like Mrs. Pac-Man because it's going in and eating up the acetylcholine. So I'm going to give her a bow and eyelashes, and she goes through and dump, 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 breaks the bond in the acetylcholine, right? So the acetylcholine goes in, or she takes up the acetylcholine and breaks it into our choline and our acetyl. And the body is really good at recycling things, so both of those things are going to go get recycled elsewhere in the body. The choline is going to go get pumped back into the neuron through a pump you need to know the name of. So the choline is going to go get pumped back in through a pump right here called the choline reuptake pump, and they just add IC on the end, so don't let that confuse you. So it's going to get recycled by the cholinergic reuptake pump right here. And that pumps the choline back into the neuron so that it can be recycled to make another acetylcholine. So here we have our choline. It goes to another chat, gets made into another acetylcholine. All right, now it's time. Let's go into the muscle cell. And we're going to see the same things that we saw in the neuron here, the same charges, the same ions. So attached to every acetylcholine receptor, I should label that for you. This is the acetylcholine receptor. Attached to every one is a tube called a T-tubule. It's actually very similar to the axon of a neuron. So that T-tubule is attached to all of them just like this. And it is attached to a structure called the sarcoplasmic reticulum. Sarcoplasmic reticulum. We're going to shorten this to SR. You may have heard of the endoplasmic reticulum, which is in all cells. Sarcoplasmic reticulum is only in muscle cells. And what it does is it contains a lot of calcium. So that calcium is all stuck in here and it's very unhappy. It doesn't want to be in here with all the other calcium because there's too much of a concentration. So it desperately wants to leave, but it can't leave until the gate opens. And I bet you can guess what charge the gate opens at because the same things are going to repeat throughout each of those. All right, so the acetylcholine binds to the acetylcholine receptor. It is a ligand-gated ion channel. So when the ligand, acetylcholine, binds to the receptor, it opens a channel for an ion, sodium. And remember that when a cell is at rest, it starts with the sodium and the calcium outside the cell. I don't know if I mentioned that in this video, but I know in class we talked about that. So at rest, all the sodium is outside the cell, right? So it really wants to go into the cell. So when that ion channel opens, all the sodium comes in. So we have sodium that enters, and it's going to travel down all these T-tubules. And if enough sodium comes in that it gets up to positive 40, you guessed it, the calcium gate opens at positive 40, same as in the neuron. But this time, that gate is on the sarcoplasmic reticulum. So the calcium gate opens at positive 40, right? Millivolts. And all that calcium that really wants to leave is going to come rushing out into the cytoplasm of the muscle cell. So all this calcium comes out. All right. Now remember what calcium does. Calcium moves things. So it's going to move those muscle filaments. So let's do a quick thing. It's just easier to show you on my fingers because it just makes more sense. So in the muscle cell, there's two types of protein. There's a thick filament and a thin filament, and they're staggered. When the muscle is relaxed, they're not overlapping, but they're staggered. And when the muscle gets a signal to contract, they slide together and overlap. So that's going to cause the muscle cell to shorten, but also thicken. Think about a muscle contracting. When a muscle contracts, it gets shorter, but also thicker. That's because those filaments are overlapping. And then when it relaxes, those filaments slide back apart, and then the muscle gets longer and thinner. This is called sliding filament theory, and the person that discovered it won the Nobel Prize for it. It's a really big deal to have discovered this. Okay, so what calcium does is calcium makes things move, and it's going to make those two filaments move. All right, so this is how it does it. There's two filaments. I don't know if I mentioned there's a thick filament and a thin filament. The thick filament is called myosin. So we're going to draw a myosin like this. And myosin has a bunch of heads on it. And these heads are actually what attach to the filament and pull it together. They're like little hands pulling a rope, right? So these heads are on the myosin. And then the thin filament is called actin. And actin looks like this, right? Thin. Actin. A-C-T-I-N, actin. All right. Now, we don't want the muscle to be contracted all the time. So when the cell is relaxed, actin is going to be covered up, protected, so to speak, by another protein called tropomyosin. So the tropomyosin covers up the actin binding sites so that the cell's not contracted. And this is tropomyosin. And then there's a fourth protein that pins the tropomyosin in place, and it actually serves also as a receptor for calcium. It's a very negative receptor, which makes sense because it wants that positive calcium to be attracted to it. It is called troponin. So troponin is going to sit on the tropomyosin like this, and it kind of pins it down in place. So when the calcium comes, it's going to go and bind to the troponin. When calcium binds to troponin, it makes it move. When the troponin moves, it moves the tropomyosin. And when the tropomyosin moves out of the way, it kind of goes like this. It moves just enough that the myosin heads are able to bind to the actin. And when it binds to the actin, it pulls it together, and that's a contraction. A muscle contraction is a muscle getting shorter, right? So every cell gets shorter, so the whole muscle gets shorter. Now, remember I said that troponin is a very negative receptor, so it's going to attract positive things to sit in it. Well, there's two other positive things. There's potassium. So we have, I'll just write it over here. You have calcium, potassium, and magnesium. Calcium and magnesium have a much more positive charge, so they're going to attach more strongly to the troponin than potassium. But what can happen is that here, I'll stand here so you can see me. The potassium and the magnesium are going to compete for that same binding site on troponin. And if potassium or magnesium is sitting there, then calcium can't bind, or as much calcium can't bind. So you get less of a contraction. And so it's called a muscle relaxer. Potassium and magnesium are called muscle relaxers. Have you ever had a cramp and your coach tells you to eat something? What do they tell you to eat? A banana, because bananas contain potassium. So what happens, let me show you. The potassium comes in, so we have our calcium, let's kind of like go back in time. So the potassium comes and sits here. And when the potassium is sitting there, the neuron sends a signal, releases the acetylcholine, which is going to activate the T tubules to open the calcium gate. The calcium goes in the cytoplasm, but it can't attach to the troponin because that seat is being taken by potassium. In reality, not every seat is taken. Some calcium binds, so you will still be able to move your muscles, but it's not as strong of a contraction. Also, it can help relieve those cramps, those muscle cramps that you get. Magnesium is even stronger because it's positive too. And so magnesium is given, for example, if a woman is going into early contractions of labor, they're going to give her magnesium because it's going to be a much stronger bond to troponin. They actually will give her something called mag, which is just magnesium, right? And that's really going to relax the muscles. And magnesium is going to be a much stronger relaxer. Okay, so let's not forget when the myosin and actin filaments are sliding together, it requires ATP. So let's write ATP so we don't forget it. Okay. Now the muscle has contracted and it's time to relax. The way that the the muscle relaxes is it pumps that calcium out of the cytoplasm back into the sarcoplasmic reticulum. It doesn't want to go in there. So it takes a lot of energy, or ATP, to pump it back in the sarcoplasmic reticulum. So we have our, on the sarcoplasmic reticulum is something called a calcium pump. The calcium pump it uses a lot of ATP to actively pump that calcium back in the sarcoplasmic reticulum. So all this calcium that's out here is going to get pumped back in. And when the calcium gets pumped off of the troponin, let me show you. Let me put my markers down. So when the calcium is pumped off of the troponin, then those muscle cells will go back to their normal relaxed state until we get the next signal for the muscle cell to contract again. Now, if you're working out and you don't have enough ATP, let's say you're doing a really long run and you run out of ATP, then your body's not going to be able to pump the calcium back in, so it's going to stay bound, so the muscle stays contracted, and that's what causes cramps. Cramping occurs. Also, when someone dies, what happens in their body is no longer making ATP. Their mitochondria aren't making ATP, so the muscles stay contracted, and that's called rigor mortis. Rigor means stiff, mortis means dead. And you can actually determine cause or time of death of a body based on how stiff it is, because it's the rate at which it has failed, the muscles have failed to relax because there's no ATP to pump the calcium back off. A couple of disorders that we're going to be talking about as we go through. I mentioned that Botox. Botox is a toxin made by a bacteria, botulism, and we have learned that that toxin affects us by cutting our snare protein. And so what we'll do is give that to people either in their facial muscles or maybe in the muscles of their lower back to alleviate back pain. And what that does is it cuts the snare so the neurotransmitter can't be released into the synapse. It never stimulates the muscle cell to contract, and the muscle stays relaxed. Another thing is Alzheimer's. People with Alzheimer's disease, we want to give them a drug that's going to increase acetylcholine in the synapse, which is going to help them remember things. It's going to help build those connections between their neurons. But when you have too much acetylcholine staying out in the synapse, it's going to keep touching the skeletal muscle cells. So as a side effect of the drug, their skeletal muscle cells keep getting stimulated, so they get tremors. So that's a side effect of Alzheimer's. Myasthenia gravis is another disease. That is an autoimmune disease that attacks this acetylcholine receptor. And so, and it specifically attacks, for some reason, the acetylcholine receptors of the muscles of the face. And so what's going to happen is as the acetylcholine receptors are destroyed, those muscles aren't able to contract. So they're going to get like drooping eyes, drooping mouth, usually only on one side of their face. That's very characteristic of the disease. And that is due to their own immune cells incorrectly attacking their own muscle cells, specifically the acetylcholine receptor on the muscle cell. Muscular dystrophy is a inherited disease. It's a genetic disease where they get a mutated copy of a protein called dystrophin. Let me see if I can explain dystrophin. Dystrophin is a protein that gives the cell, gives the cell structure and shape. It holds it so it's a nice oval, just like this. And it's also going to prevent anything from leaking out of it, like plugs it up. A mutated copy of dystrophin in people that have muscular dystrophy, what happens is their cells don't have that same structure, so they begin to kind of collapse. All their skeletal muscles begin to collapse at a cellular level, and it creates these little micro holes where it's going to leak out all these important ions that they need. So that's why in muscular dystrophy, over time, they're unable to use their skeletal muscles, obviously, and so over time they lose the ability to walk, to move, and eventually it just progresses. It's a progressive disease. Okay, hope this helped. Bye, guys.